Today we are going to be talking about the triple sugar ion test, that is the TSI test. So let us see the introduction of this TSI test. Some bacteria have the ability to ferment carbohydrates, particularly the sugars, either glucose, sucrose, or lactose. And among them, each bacteria they can ferment only some of the sugar, while it cannot ferment the other. Thus, the sugars which a bacteria can ferment and the sugars which it cannot is the characteristics of the bacteria, and thus it is an important criterion for its identification. So the TSI is basically a microbiological test named for its ability to test a microorganism which has the ability to ferment sugar and produce hydrogen sulfide. So coming to the principle, this TSI is a test that is designed to differentiate among organisms based on the difference in carbohydrate fermentation patterns and hydrogen sulfide production. So this carbohydrate fermentation is indicated by the production of gas and change in the color of the pH indicator from red to yellow. In a simple way, let me also tell you in another way the principle. This phenol red is for detecting the carbohydrate fermentation, and ferrous ammonium sulfate for detection of hydrogen sulfide production. So here the carbohydrate fermentation is detected by presence of gas and a visible color change from red to yellow by the pH indicator phenol red. Then the production of hydrogen sulfide is indicated by the presence of precipitate that blackens the medium at the bottom. Now coming to the media, here we are going to use the TSI agar. So TSI agar is it is a mixture of a different components. So here I have given the constituent of the TSI agar. Now coming to the procedure, the first thing you have to do is that with a straight inoculation needle, you have to touch the top of a well isolated colonies. You can pick up the single colonies, depends upon how the colonies are in your plate. Then you have to inoculate the TSI by first tapping through the center of the medium to the bottom of the tube, and then streak gently on the surface of the agar slant. Remember, it is a step and streak. Here in the TSI method, you are going to step and streak, whereas in citrate you are going to streak only on the slant. Please don't get confused with that. Then you have to lift the cap loosely and incubate the tube at 35 to 37 degrees centigrade in ambient air for 18 to 24 hours. Then after 18 to 24 hours, we have to examine the reaction by the reaction on the medium. So here in the interpretation, I have given the image. I have given the image for the interpretation. So the first one, the gas production. So what is this gas production? The first, here in the yellow tube, you can see there is space which is usually a gas production, a bubble or a crack. There is a bubble or kind of a crack in the agar. This indicates that there is a production of gas, the formation of carbon dioxide and the H2, hydrogen sulfide. So the medium has a bubble or a crack in the medium in the middle when it was incubated at 24 to 48 hours. Now the next one we have here is an acid by acid, which means yellow slant by yellow butt reaction. This means that the fermentation of a dextrose, lactose, or a sucrose take place. That is why the reaction has a yellow slant as well as a yellow butt. Now the next one in the third tube, you have an alkaline, and as you can see here clearly, you have a red slant, then the yellow butt. This is because there is an red slant and yellow bud. This is an indicative of dextrose fermentation only. This means that only one kind of a sugar has fermented, only one kind of sugar fermentation take place. That is why you have got a red slant and a yellow bud. Now the fourth one, there is a blackening of the medium. This occurs because of the presence of the hydrogen sulfide. When since there is a precipitate that blackens the medium at the bottom, this is because of the hydrogen sulfide. Now the last tube we have here is red. All the tubes are all the slant as well as the bud is red, just the opposite of the second tube. In second tube it is yellow by yellow. This means that means that there is a fermentation of either dextrose or sucrose or lactose. Now in case of the last tube, here we have red slant by red bud, which means that there is an absence of carbohydrate fermentation, so none of the sugar fermented in this case. So this is all about the TSI test.